மேடம் இப்ப பாருங்க
சார் யூடியூப் ஆன்ல தான் சார் இருக்கு, இன்னும் லைவ் ஸ்டார்ட் ஆகலங்க சார்.
ஸ்டார்ட் ஆயிடுச்சு பாருங்க, ரிஃப்ரெஷ் பண்ணி பாருங்க.
சார் லைவ் வந்துருச்சு சார். லைவ் வந்துருச்சு சார். மேம் வந்துருச்சு மேம்.
சாட் பண்ணி பாருங்க.
இல்ல எனக்கு இன்னும் வருது சார், வருது.
லைவ் வந்துருச்சு சார்.
வருது சார். ஓகே.
சாட் பண்ணி பாருங்க.
இல்ல எனக்கு இன்னும் வருது சார், வருது.
Good morning, madam. Yes, bro, sir. Good morning. Okay.
சார் இன்னும் எனக்கு chat இது வந்து ஓபன் ஆகல சார்.
A very good morning, dear participants. I extend a hearty welcome to our resource person.
First open பண்ணுங்க.
இத மட்டும் நீங்க ஆஃப் பண்ணி விட்டுட்டு இந்த சிம்பிள் இருக்குல பாரு, இந்த வீடியோ ஆஃப் பண்ணிட்டீங்கன்னா நீங்க தெரிய மாட்டீங்க. இது ஆஃப் ஓகே. இத மட்டும் ஆஃப் பண்ணி விட்டுட்டு
சார் 1 செகண்ட் சார், திருப்பி ரெஃப்ரெஷ் பண்ணிட்டு இருக்கேன் சார், வரல. எனக்கு விஸ்காமோட கனெக்ட் ஆயிடுச்சு சார்.
இத மட்டும் ஆஃப் பண்ணி
சார் வந்துச்சு சார். 1 செகண்ட் சார், திருப்பி ரெஃப்ரெஷ் பண்ணிட்டு இருக்கேன் சார், வரல. எனக்கு விஸ்காமோட கனெக்ட் ஆயிடுச்சு சார். சார் வந்துச்சு சார். 1 செகண்ட் சார், திருப்பி ரெஃப்ரெஷ் பண்ணிட்டு இருக்கேன் சார், வரல. எனக்கு விஸ்காமோட கனெக்ட் ஆயிடுச்சு சார். சார் வந்துச்சு சார். அகைன் எனக்கு chat ஆப்ஷன் ப்ராப்ளம் இருக்கு சார். சார் அகைன் எனக்கு chat ஆப்ஷன் ப்ராப்ளம் இருக்கு சார். சார்
சொல்லுங்க மேடம்.
சார் chat ஆப்ஷன் எனக்கு இன்னும் வரல சார். லைவ் chat போனாலும் வரல சார். எனக்கு மெசேजेस வந்து சென்ட் பண்ண முடியல. ரெண்டு ஆப்ஷன் தான் சார் கேக்குது. ஒன்னு வந்து create a channel to join the chat, அதுதான் சார் வருது.
இங்க வரணுமே எல்லாரும் வந்து வேற யாராவது ட்ரை பண்ண சொல்லுங்க.
இது கேரளால போறோம்.
சார் chat மெசேजेस என்னால வியூ பண்ண முடியுது சார், பட் என்னால எந்த மெசேஜ்மே போட முடியல சார், எனக்கு வரல.
சரி, இந்த செஷன் ஆரம்பிச்சிட்டு என்னன்னு பாப்போம்.
ஓகே சார். சார் அப்படி இல்லைன்னாலும் அந்த லிங்க் மட்டும் தானே சார் சென்ட் பண்ணோம். லிங்க் வேணா கார்த்தியை வேணா போட சொல்லிடலாம் சார் இந்த இதுல.
சரி.
ஓகே சார்.
கார்த்தியில சொல்லி போஸ்ட் பண்ண ஏதாவது மெசேஜ் போட சொல்லுங்க, சேட் பண்ண சொல்லுங்க.
கார்த்தி
மெசேஜ் போட முடியுது சார், மெசேஜ் போட முடியுது சார்.
நம்ம எப்படி அவங்களும் பண்ண முடியுமே, அவங்களுக்கு ஏன் வரல?
இல்ல நிறைய பேர் chat boxல போட்டு
மேம் போட்டேன், மெசேஜ் போட்டேன் பாருங்க, வந்திருக்கும் chat boxல.
இல்ல அது மெசேजेस என்னால வியூ பண்ண முடியுது, பட் எனக்கு எந்த மெசேஜ்மே போட முடியலையே. இந்த வந்திருக்கு மெசேஜ் பார்க்க முடியுது.
நீங்க ஜிமெயில் அக்கவுண்ட்?
ஆமா சார், ஜிமெயில் அக்கவுண்ட் தான் சார்.
அப்பனா வரணுமே, கண்டிப்பா வரும். சரி என்னன்னு பாத்துருவோம், டைம் ஆயிடுச்சு.
ஓகே சார். ஓகே சார், we can start. நம்ம அந்த லிங்க் மட்டும் கார்த்தியை போட சொல்லிடலாம் சார்.
ஓகே ஒண்ணும் பிரச்சனை இல்ல.
ஓகே சார்.
சார் நான் பேசுறது ஆடிபிளா இருக்கா?
சார் கேக்குது சார்.
பண்ணியா சரி கேக்குது.
காணிக்கிறார் சார், என்னோடது கிளியரா இருக்கா?
இருக்கு சார், நல்லாவே இருக்கு.
வீடியோ கிளியரா இருக்கா?
இருக்கு சார்.
ஓகே, ரைட் ரைட்.
சார் என் வாய்ஸ் கேக்குதா கிளியர்?
கேக்குது சார்.
நல்லா கேக்கு, நல்லா
ஓகே.
நல்லா இருக்கு. ராஜன் சார் வீடியோ ஆன் பண்ணாருன்னா ஆரம்பிச்சிடலாம். ராஜன் சார் உங்க வீடியோ ஆன் பண்ணுங்க.
ஆன் பண்ணிட்டோம். இது சவுண்ட் ஃபேன் சொல்லுவாங்க.
அப்புறம்
காணிக்கை சார் கேக்குதா?
சார் ஒரு நிமிஷம் இருங்க.
சத்தா இருக்கு. கேக்குதா சார்? கேக்குதா சார்? கேன் யூ ஹியர் மீ? ஓகே சார். ஓகே, thank you. No problem. Take your time. Just want to check.
Screen வருதா?
We can start now.
Yeah, sir. Yeah, sir. Good evening to one and all gathered here, and thank you for joining us today for this webinar on microbes, the drivers of human life. At this time, I invite our resource person, Dr. G. Velmurugan, scientist, KMCH Research Foundation, Rector Father, Reverend Father Leonard Fernando S.J., Dean School of Biological Sciences, Head Department of Botany, faculties of SJC, and participants to come forward and begin our session. It is a mark of our undying tradition to invoke the Almighty at the beginning of an important event. Like the great philosopher Søren Kierkegaard said, the function of prayer is not to influence God, but rather to change the nature of one who prays. Let us all remain silent for the prayer song.
Deliver us, Lord, from all that is evil, and grant us peace in our day. In your mercy, keep us free from sin and protect us from all anxiety, as we wait in joyful hope for the coming of our Savior, Jesus Christ.
Hospitality is inarguably one of the defining qualities of St. Joseph College. May I now invite Professor S.R. Sendil Kumar, Head Department of Botany, to welcome the gathering.
Greetings in His holy name. Very good evening to all of you. The recent developments in the field of bioscience research is to develop multidisciplinary approaches and exploit emerging technologies in underpin improvements in both human and animal health, and also promote effective translation of bioscience to meet the key challenges of enabling people to live healthier lives for longer and to support innovation. This way, the Department of Botany is sparked to organize a webinar today on microbes, the drivers of human life. Provides an opportunity to interact with the subject expert. Here, Reverend Father Dr. Leo S.J., rector of this great 176 years old, very prestigious Jesuit institutions of South India, is known for his persistence, devotion, and desire to serve with the Jesuit vision and mission. Father is our successful administrator with a keen presence of mind, with unflinching courage. Father is a spiritual leader and simple to the core. On the behalf of the botany department and on my own behalf, I bid a hearty welcome you, Father. One of our alumnus, Dr. G. Velmurugan, scientist, KMCH Research Foundation, Coimbatore, in South India, who is the speaker of today's webinar, is here. He will be introduced later in detail by Dr. K. Rajan. On the behalf of the botany department, on own behalf, I extend my heart welcome to Dr. Velmurugan. Special note of welcome to Dr. S. Sagaya Sadish, Dean, School of Biological Sciences of St. Joseph's College, Tiruchirappalli, and the organizers of the webinar of lectures, Dr. K. Rajan, Associate Professor, and Dr. S. Anusuya, Assistant Professor, Department of Botany, St. Joseph's College, Tiruchirappalli. I also welcome our department colleagues in a special way. Finally, I welcome all the webinar participants of various institutions from different parts of our country and abroad from Saudi Arabia, Iran, Iraq, Philippines, Hong Kong, and other few East Asian countries. I once again welcome one and all. Thank you.
Thank you, sir, for your kind words of welcome. Now I request Reverend Father Leonard Fernando S.J., Rector, SJC Trichy, to felicitate the gathering.
Good evening to you all. I'm told around more than 1,000 people are taking part in this webinar. And I'm all the more happy also to know that the speaker of the day, the resource person, is our own alumnus of the college. One of our illustrious alumni coming over to give this webinar talk. I greet all of you who are taking part in this webinar organized by Botany Department of St. Joseph's College on the topic, Microbes, the Drivers of Human Life. I'd like to take this opportunity also to congratulate the organizing secretaries, Dr. K. Rajan, Associate Professor, Dr. Anusuya, Assistant Professor at St. Joseph's College, Trichy, Dr. Sahai Satish, Dean of School of Biological Sciences, and Dr. Senthil Kumar, Head Department of Botany of St. Joseph's College. When I browsed through the topic, I found it very interesting and captivating. Even to me, who is not a botany student, but someone who is touched or influenced by what is going on around in the world today because of these two areas of the COVID-19 pandemic and the recent one of the locust swarms. And it is in that context, I think, we see so many of us gathering together to listen to Dr. Velmurugan, a scientist from Coimbatore, KMCH Research Foundation. At this time of great anxiety, a webinar, I think, is very important because through this webinar, we are going to hear the pivotal role played by microbes, the drivers of human life, plant life, and animal life. And much more so when I read the abstract from the scientist or alumnus, illustrious alumnus, that we'll be focusing on the many positive things that the microbes give to us, not only the negative things that today very, very much is spoken about the negative things. So at this moment, I think it's a very innovative and enriching topic Dr. Velmurugan, you are going to give to us, and we are looking forward to it. And I welcome you and greet everyone, and God bless us all. Thank you so much. Welcome, Doctor.
Like the rainwater bringing life and prosperity to the land, your words bring enthusiasm and strength in our hearts. Thank you, Father, for your supportive words of appreciation. Now I request Dr. Rajan, Associate Professor of Zoology, St. Joseph College, to introduce the resource person, Dr. M. Velmurugan.
Hello and welcome to all the participants. It is my pleasant duty to introduce our resource person, Dr. G. Velmurugan, for today's webinar on the topic Microbes, the Drives of the Human Life. His academic and research achievements are vast and run more than 5 pages. Due to want of time, I am pleased to brief it. At present, he is working as a scientist in KMCH Research Foundations, Kovai Medical Center, Hospital, Coimbatore, Tamil Nadu, South India. Since he is an illustrious alumnus to our St. Joseph's College, obtained his undergraduate degree in 2005 with his distinctions. Then he did his MSc and PhD in Madurai Kamaraj University in 2005 to 7 and 2011 to 15 respectively. So due to his meritorious, extraordinary academic and research achievement, he received many awards and stood different positions. So here at the highlighting Indian Academy of Science Foundation's Research Fellowship 2006 and International Max Planck Research Fellowship 2008 from Germany, and Biological Science Meritorious Students Research Fellowship in 2013, and Senior Research Fellow 2014, Postdoctoral Fellowship 2016, and Young Scientist Award 2017, and Tamil Nadu State Level Eligibility Test for Assistant Professor in 2018, that is SLIT, and earlier Career Scientist Travel Award in 2018, and Travel Bursary Award in 2019, and International Travel Grant Award in 2019 from CSIR. So thus he is having an enormous amount of research capacities, and the meaningful publications is 14 published manuscript and international and national level, published and invited book chapters in 2 in Springers and elsewhere. As well as he has an H index of 8, an I-10 index of 8, and cumulative impact factor is 73.8. It is very imaginable. So you can see how he is well potent in his transformation of research into meaningful findings. And the total citation is 289. So average here, the first authored publication is 81.3%, and he is co-authored in 44%. Based on his research, he is mainly undergoing various activities as well as research capacities in difference. So a popular artist, that is the articles published in various journals as well as the dailies and radio news. Now, based on his project work, that is the principal investigator, nearly 6 ongoing projects are going, as well as a co-authored for 3. So a tune of 2 crores, 75 lakhs more rupees have been claimed by his research from various agencies, including DST, ICMR, as well as UGC and KMC Research Foundations. And he is now positioned in various capacities as research fellows, research technicians, CSR research fellows, and Institute of Postdoctoral Fellow in various activities of his research, and as well as Institute of Postdoctoral Fellow by the external scientist in KMC. So there is the, now he is membership in different Association of Microbiologists in India, Aquaterra Society for Biological Science Life members. And he is delivered more than 8 invited talks in different universities as well as in abroad, South Korea. As well as he is publishing many books, there is the chaptered in elsewhere, etc. So we have an eminent and simple and humble research and resourceful personality with us. So we are very proud and happy to have you as a resource person for today's webinar. And moreover, we would love to hear from you during these presentations. Thank you so much. Over to the speaker, Dr. Vel Murugan. Now you can take over the sessions.
Okay. Thank you. Thank you, thank you, Father, and thank you, my teachers. It's really the organizers are my teachers who developed me really. So I will just share my screen. So please make sure you are able to sit visible. So my screen is visible. Hello?
Yeah.
Are you able to see my screen?
Yes, sir. Yes, sir.
Hope everybody is able to see my screen. No. I've shared my screen. Yes. Okay. Okay. Thank you. Now everybody is able to see my screen. Fine. Okay. So before going into my talk, I would like to say why I was here. So it was a really proud moment, as Father and others said. Saying I am a Josephite is a proud thing. When I was studying, we celebrated 160 years of St. Joseph's College, and now I completed in 2005. Now we have reached 176 years, and even when I go abroad or anywhere, it's always a proud moment to say I am from St. Joseph's College. Even still, I remember when I was studying, I always used to enjoy this by saying to my students and everybody, when I was doing my BSc Botany during 2002 to 2005, the President of India, the Chief Justice of India, the Chief Election Commissioner of India, all the three are from my college. So it's been a proud moment during that time, still date. So I think it's a rare occasion. So I was there in the college during that occasion when three top units of the India were from St. Joseph's College. And also I want to thank all my teachers during that period. Everybody has played a key role in building my career. Still it takes more time. I want to say everybody's name. Dr. Abdul Kader, Dr. Patrick Gomez, Dr. Irude Arogyaswamy, Dr. Melchias, Dr. Jayachandran, Dr. John, Dr. Sendil Kumar, Dr. Rajan, Dr. Sadish, and Dr. Selwyn. So everybody has played a key role in my career. Everybody taught me a new vision and which takes me the next directions. Especially when I speak about my career, I used to say who I am was shaped in different institutions, and the BSc Botany in St. Joseph's College has developed an interest on ecology and environment. Today, I am not working with botany or in a plant subject, but still, though I am working in a hospital and on a human health, still proudly I can say I am working on ecology and environment. Still I am looking into the ecological and environmental factors, how it affects the human health. Even many people have tried me to get out of this, but I being very stubborn that I will do only research which is related to environment. So that love and care for environment was seeded in me during my days in doing BSc Botany at St. Joseph's College. So I am very thankful to the institution for this, that shaped my career and which took on me. So coming to my topic. So as I have to thank the Father who summarized my talk nicely. So what I'm going to present. So first, so I will have made the talk in a basic way. So that since the students are also there, they will also have a understanding. So we will speak about the basic things and in an elaborate manner what is the importance of microbes, and then we will go specifically to currently major issues like COVID-19 and the locust swarms, how microbes play a role in this. So who are microbes? How you will define microbes? So microbes are essentially, so as the term says, they are microscopic. So they are not visible to the naked eye. So they range from different organisms. They are largely unicellular like bacteria, protozoa, fungi. They include these fungi and algae would be from the plant kingdom. Protozoa is from the animals. And bacteria and viruses are more ancestral, and they are called as prokaryotes who does have a defined organ system. And even viruses which include this coronavirus and other disease causing agents. So these viruses are, even we can't define it whether they are living organism or non-living organisms. They are like a non-living particle when they are staying living system. When they enter a system only, they gain the life, they replicate and they do. Otherwise, they are just a protein particle and a nucleic acid engulfed in just a biological material. That's it. It doesn't have a life. But when it enters, even there are different kinds of viruses. Not only the viruses cause diseases. Even there are viruses which kills bacteria. Even there are viruses which lives within us. Even I will come with everything. So these are the size of the microbes. As I said, this is the range of the molecules which ranges from nanometer to meters, you can see. So these are what people, so these are very sub-nanometer, which are organs and different protein molecules. And these which are shown in red color are the microbes. So they include viruses which range in around 10 to 100 nanometers. And in microns, bacteria, fungi, unicellular organisms are included. So they are very tiny, and we need high resolution microscopes to see them. Okay. So they are plenty. They are diverse. They are plenty. There are many species, many families, and there are many shapes. Even they change their shapes. They adapt to every environment. They are very colorful. Even if I make a map of, even I remember Sendil sir used to tell me orchids are the most beautiful flowers. So when I make a picturization of all bacteria and different viruses, so these are the different viruses, and this is a bacteriophage. They are more beautiful. Even they are invisible, but when we see, they will be more beautiful. And one more thing is they are not all harmful. So that's one of the major thing I want to stress today, as nicely mentioned by the father. I want the real objective of my talk is to say not all microbes are harmful. So where are they? So I said there are plenty of them. There are different types. Where are they? So they are really omnipresent. They are everywhere. Even if father don't mistake, people used to say God is everywhere. I don't know whether God is everywhere or not, but I'm sure these microbes are everywhere. They are present in all the spheres of the world, including even in now data from NASA and other agencies have shown even the microbes are present in extraterrestrial space also. They are present in air, in all organisms, in rock, in water, in deep sea sediments of the sea. Everywhere it is present. So these are the major habitats of the microbes. So actually, the majority of the microbes are present inside the sea. So even this is largely unexplored. These were made using some mathematical calculations. These numbers have been made. It's not really not exactly calculated by some stimulations. They have calculated how much it could be. But a large amount of the microbes are residing inside the sea and below the earth. And they are doing a lot of work, and they are the drivers of the whole world. Even I initially sent the title as drivers of human life. But later I recognized I should have mentioned as drivers of the world. That should be the correct title. So rather than saying just humans, they are the drivers of the whole world. They regulate the atmosphere, they regulate the carbon emissions, they regulate everything. So you can see they associate with all forms of biosphere and all forms of life. So if you take plant kingdom, right from starting from algae to bryophytes, gymnosomes, pteridophytes, angiosperms, they associate with everybody. Similarly, in case of animals also, starting from poriferous sponges, annelids, insects, molluscans, mammals, they live in everybody. So this one character we have to learn from microbes. So we have to learn how to live in this world along with other organisms. We can't live in this world by just making extinct another organism, and human cannot prosperously live. That is what we are in the last few centuries, what we have done to the environment in terms of industrial development, pollution, and many, many things. What we have created as we made several mistakes. And this thing we have to learn from microbes, that we should learn to adapt to live with every creatures in this world. And really the microbes are really doing that. They are living with everybody from the lowest taxonomy class to the highest taxonomy class in plants, and also and even if it is take to come to the plants, they live in the soil, they are in the roots, they are inside the plant tissues which are called as endophytic microbiome. So the microbiome is a holistic word which contains all the different kinds of microbes in a system. So phylosphere refers to the microbes which are present on the leaf surface, and they play a very major role. They not only, even there are some microbes which causes diseases in the plants, but without the microbes, the plants cannot survive. They need them for all their activities. So I will come to that later. So first I will explain where are they. So they are in everywhere. So soil. And when I speak about, as a botany student, I had ancillary zoology. So when I just learn about these sponges, for those who are unfamiliar with sponges, they are in the second phylum of in the animal kingdom, which are called porifera, and they are motile organisms or non-motile organisms. They are animals, but they look like a plant and they cannot move. Just the water flows through one way and it comes through other way. These are all I learned during my botany allied zoology classes, especially from Rajan sir. So once the sponges or marine sponges are numerous, they are fluorescent, they do multiple activities, and several medical beneficial activities has been known. Metabolites have been isolated, which produces treatment for different diseases has been reported. But now it is known that 50% of the biomass, if you weigh a sponge, the 50% of the weight of that sponge is contributed by the microbes which are residing inside us or which are traveling along with the water. So you can see these triangles, these circles represent different microbes. So some microbes just, so they have numerous pores all through the body through which the water enters. So through the water, they take the nutrients. Not only the nutrients, they have microbes and many metabolites which were around 20-30 years, which were identified to be produced by microbes, are today proven they are really not produced by microbes, by the sponges, but by the microbes. So now the sponge has been termed as a microbial fermenter. So fermenter is an industrial device which is used to culture the microbes in a large scale and to produce some material of interest from the microbe. So now this is a natural fermenter which plays a key role in the thing. So microbes is, as I said, even in sponges are there, even in insects they are there. Even in insects, in salivary glands, it is there. Even in the so intestine is which is a part of the stomach digestive system is a major. Which is a major location where there is more microbes are concentrated, and they plays a key role in digestion and numerous responses in the body. So even in insects they are residing, and humans. So as being a human, everybody will be interested to know what is happening in humans. Now whatever the medical biology and whatever treatments we are doing is based on what we learned, how these organs are working. So all the doctors and everybody are treating us by just understanding what these organs are doing. But in reality, we have millions of microbes residing within our human body in all the places. A few years back, it was known only intestine and oral mouth and skin microbes are residing. Now it is getting proven even your liver has bacteria is living, even in cancer tissues, microbiota, new bacteria are living, and it's a complex system where, and they send signals. So the bacteria in the stomach sends signal to the brain. Now it's shown a person getting anxiety or a person getting mentally disorder is also controlled by the bacteria which are residing in our intestine, and it varies based on our day-to-day habits, what we eat, to what we are getting exposed, and different parameters determines European having a different food habit have a different microbes in their gut, and we have South Indians have a different microbe composition. So it varies from people to people. So to understand that is why maybe we are not able to tackle the coronavirus or many diseases in reality. We don't have a cure for we are able to control the diseases only. We are not able to really cure the diseases, whether it is diabetes or cancer. These microbes are not only playing a role in so-called infectious diseases, the diseases which are caused by microorganisms, they are also playing a role in all kinds of diseases, which we call it as non-communicable diseases, which cannot spread from a person to person, like diabetes or hypertension, heart disease or a liver problem. Even in those diseases also, these microbes are playing a bigger role, which we not studied, which we not understand, and we have now just now started. So after understanding the complete thing only, we can give a right medicine to a person. So as I said, this is the microbes. So as you know, all the organisms are made of cells, right? So the number of cells in the human body is just 100 trillion. But the number of genes, if you see, there are only 30 trillion cells in a human body, but there are 100 trillion microbial cells within the human body. So just a part, when you just take a proportion, just a part of the leg, if you put all microbial cells in one part of the body, it would be just part of the leg. And in terms of genes, there are, in comparatively, it is just a part of your food. The human genes are there, just 23,000, and whereas 99,000 of different microbial genes are there. There are a lot of studies have been done in the last few decades on human genome, how it varies, what role it plays in diseases and different things. But what we have studied, what we have done is just this part of the human body. So if you put all the genes in one region of the human body, it could be just this size. But we have a lot of things which are residing within us, which plays a major role, both beneficial as well as sometimes when you disturb them, they will give a feedback to you. So I will explain it later. So they are present everywhere. Just it's a percentage, it is graphically represented. So even for body weight, it contributes, the bacteria contributes nearly 2 to 3%. In terms of weight, it could be very small, but just a 3%. So if you are so the 100 kg body weight, just a 3 to 4%. But if you take number of cells, it was 8% weight of number of cells only from human system. So blue color is the human system, whereas nearly 90% of the cells are from microbial system. And in case of genes, it is just less than 1% from the human system, and 99% are from the microbes. So it's not only just the human system. Just for an example, I have shown the human system, and as we are humans, we studied more about humans and we have more data about humans. Even all the organisms, plants, microbes, everything has this much microbes. So what can we isolate and grow all? So before going into what they are really doing, so now, as Sindhu sir has rightly pointed out, now technologies have developed. So as we actually still we don't have the knowledge to isolate and grow everybody. So humans can isolate and grow only at 10%. So this is the classical microbiological technique in which we take some samples, a soil sample or a fecal sample or from insect, and we just put in some nutrient media, and then you put it in a petri dish and you see what are growing. But we do in a condition where it was largely maintained in 37 degree and in some nutrient conditions. But we don't know how to grow more than 90% of the microbes. But today we know what microbes are there. How we know is we are directly extracting the DNA because we don't know how to isolate them. So we are directly taking the genetic material, and by next generation sequencing combined with the developments in computational science and statistics, mathematics and other fields, now we are able to identify what are the different microbes are there. Even we don't know how to grow, but now we are able to say who is there. Now it is possible. So that is called the unculture, which we largely call it as metagenomics. So through this now, large number of new species have been identified. And so as I said, they are everywhere. I said it is omnipresent, right? So they are present in extraterrestrial space. They are present in soil, plants, animals, in air, everywhere. What are they doing really? Okay. So what are they doing? As I said, they cause diseases, right? But we are not all bad. I join with them. So I will speak on behalf of them. So we are not all bad. Okay. We are really helpful to you. We created this world. We are your ancestors, and we created everything. We only made this world. We created this earth crust. We created the mountains. We dissolved the mountain into soil, and we made the soil fertile. So we are the creators of this world. We don't know whether these microbes are God or not, but they are somewhat, they created the base for the plant and animal life, and still they are doing that. It is not just they did and left. They are still living with us, and still they are doing their duties. So as I said, they do a lot of work. on every system they do. You can see even today they are the major key players in global climate change. Also, if people heard about carbon dioxide release in the atmosphere, greenhouse gases, and the microbes which are residing in the deep seas plays a major role in sequestration of carbon dioxide, and they play a role in microbial metabolism of different metabolites. Even the sea, the microalgae which are in the sea plays a major role in the global production by means of photosynthesis, which acts as food for the fish and fish beings. So maybe we're not directly consuming the microbes, but microbes are playing a key role for our everyday life, and they not only helpful to feed us, even they are the players which destroy the dead bodies, they destroy the waste materials, and they place everywhere, and they converts the dead materials into fossil fuels from which we generate electricity. So everywhere it is there. So they play a key role in every process. It is not just biology. Without microbes, there will be nothing on this earth. Okay, so without microbes, there will be no rock, no soil, there will be no computer science, I can say, there will be no commerce, you can't do any business, because without these resources, there won't be anything you to sell and market. So these are the major ones, they play a key role. So in case of plants, they, as I say, they reside in the root and the leaves. They help in everything. We call it as PGP, those bacteria which help. We call it as plant growth promoting rhizobacteria. They help in. They produce some hormones. They help in taking iron from the soil. They play a major role in taking phosphates from the soil. Now we have killed them. That is what I always, even that is what I was taught in during my BSc in the term of organic farming. That is what I was taught by my teachers during my duration in St. Joseph College. In the name of global farming green revolution, we have introduced a lot of phosphate fertilizers, many things into the soil, and they really killed them. Still, I am working with that. I am still studying on, I will come to that later part, how these chemical fertilizers and pesticides are, how they are destroying every system and including human health, and why that's I'm going to discuss that, and what's the role that the microbes are playing. Everything we are going to discuss. They play a major role. Not only they are just residing on the surface, even fungi and some bacteria, they call it form of mycorrhizal association. And even during my period in Germany, initially I started my work with plant microbe interaction. I worked on these microbes, and then I was working on mycorrhiza, and then I moved to animal microbe interaction. So still I was doing the same, just the system varies. I was working with microbes, I can say. I was working with microbial ecology. I was working with how the microbes metabolizes the things. That's what I am continuing. So this microbial balance is very much essential. Whatever, sometimes foolish things like introduction of any material, any new contaminants into the environment, whether it is in the environment or inside our human body, it makes an imbalance. It destroys the ecology, the biodiversity. When we speak about biodiversity, everybody speaks about deforestation, extinct animals and everything. Biodiversity exists everywhere. Even within a human body, there is a huge biodiversity. If you destroy that, that's result in disease. Even many doctors who are treating us, even I know physicians who ask me, what is microbiota? Even they don't have, even their syllabus, when they study microbiology, they study them as bacteria and virus and diseases. They don't know there are there are plenty of microbes residing within us, which plays a major role, and that causes the disease. Even in case of many diseases in Tirukural, there is a kural, Noi Nadi, Noi Mudal Nadi. Before treating a disease, the physician should know what is the disease, what is the root cause of the disease. But in real clinical practice, there is largely, we don't know what is the root cause of the disease. Without knowing the root cause of the disease, we are just trying to treat the disease. So maybe the microbes could be one among the factors which would be a root cause for the disease. So they are everywhere. Even they determines the action of chemicals. Some medicines are working for somebody. Even if you take this corona disease, they say some drugs, some antiviral drug, Admicera, hydroxychloroquine, queens. It's working for some set of people, and some people it making some critical conditions which leading to death, and still we are not able to find a correct drug, and for some people without any treatment they are getting recovered. So already there are many investigations which have shown that the microbiota plays a key role, and they make some drugs which are not active as inactive, and they make some drugs as a toxic substance which makes some people get some side effects when they take a drug, but it is not the same for the other people. So they do a lot of things, and they plays a key role even in this drug, and not only drugs, even with environmental pollutants. So this is my PhD work where I have shown that pesticides are inducing diabetes. So this work was, it's a kind of where I got inspiration. So whenever I speak in some way I will get into speaking about my college. So shepherd activity, where we use every class we used to adopt a village for 2.5 years, and we go visit that village every time, and we divide our class itself into different groups like education, farming, and we help the farmers, we help them to get touch with the agriculture officers, and on different strata we help the people, and there are different villages which are adapted under the shepherd program. So when I was doing my PhD, we just made a survey on prevalence of diabetes in the villages around Madurai Kamaraj University, and generally diabetes or any other disease like heart disease are considered as urbanized disease, which are due to high fat diet, cholesterol, physically who are not working, just sitting and doing some computational work. So these are the risk factors generally considered for diabetes. But surprisingly, we find threefold higher prevalence of diabetes among farmers when we compared with the non-farming people from the same village. And that it was really shocking for us, because generally even the doctors think that villagers don't get these diseases, but we found they are getting these disease, and by a lot of experience. I'm not speaking much about this work today, but I will just say a summary of this. So you can see these black stars indicates the insecticide. So when it enters our body, it goes to the intestine. In the intestine, the microbes which are present in the intestine converts this OP is a type of insecticide which is largely used. It is called organophosphate, and it converts into some chemical substrate which is being converted to glucose by our human system. So they cause diabetes. Even I have wrote an article in a news in which we said the microbes say if you kill me with insecticides, I will kill you with diabetes. So it's like saying in Tamil, நான் நல்லவனுக்கு நல்லவன் கெட்டவனுக்கு கெட்டவங்கிற மாதிரி நல்ல சாப்பாடு போட்டா நான் உனக்கு உதவி செய்வேன். If you give me toxic substances, then I will also reciprocate in giving you diseases. So this we have proved in animal system and even in human system, and it was published in Genome Biology, and it has been cited in Hindu, NDTV, and internationally in many news journals. So not only these pesticides, even other toxic pollutants. So this bisphenol A, saccharin is an artificial sweetener which is present in all soft drinks like Coca-Cola, Fanta, and all those things. Even in fruit juices also now people are using. Even it was promoted as alternative to, it is called as non-caloric sweeteners. Even it was promoted as an alternative for the diabetes patients. But there are strong data proven by different people that they also cause diabetes. But in reality, we are seeing day-to-day different advertisements. Even the doctors are recommending to use these artificial sweeteners. And these microbiota plays a role. They convert into just like metabolizing the pesticide, they metabolize them and they are causing diabetes. So not only the pesticides, the bisphenol A is a component which are present in plastics, and emulsifiers are being used in different cakes and food substances, and even toxic heavy metals are being metabolized and they are causing diabetes. So this intermediate layer, so these are the intestinal cells where the microbes are residing. So whatever we eat reach them and they convert them into what it is into some other metabolites. For example, in this case, it is converted to here, and in many cases, still it is not known how they induce the disease, but it's known when the microbes are not present, they are not able to induce the disease. So there is a lot of studies, and now we have got an Indo-French project to study on collaboration how still we are not sure what microbes are involved. We just know the mechanism, whether that microbe itself can become a drug. So we are working on that. So based on this, now we have proposed this, that gut microbiota should be included in toxicological risk assessment. So toxicological risk assessment is a technical protocol. When a new drug or a new chemical is being introduced, they will do some basic tests to make sure it is not harmful for the human cells or for the environment or like that. But nobody studies whether the drug or chemical is disturbing the microbiota. So we have made this suggestion, and it was presented in an international toxicological conference, where we have proposed that this should be gut microbiota, not only gut microbiota. That conference was concentrated majorly on human health, so we mentioned about gut microbiota, but there should be a risk assessment protocol should be developed for any new drug or any new chemical which are being introduced into the environment or the human system, to make sure that it not disturbs the microbial ecology. So as I said, ecology is not everywhere, not it just to UNESCO heritage site like Western Ghats or the forest, Amazon forest. There is ecology, there is a huge diversity, even in your hand, even in your skin, there is a huge diversity. So we are destroying everywhere. I will come to that. That's one of the major point I want to make it today. So now I have described, so as I said, they drives everything, not only the human life, they drives the whole world. They plays a major role in agriculture. They play everywhere. So they do plenty of work. Even we know a little. Really, we know maybe a 0.1% of what is microbes are doing. 99% we don't know what they are really doing, staying with us or staying in the deep oceans. They are large. Even they are residing even in the volcanoes. Hope everybody of you have been biology students will be familiar with PCR. So I always say basic research is important. When you do PCR, you do at a higher temperature, and for that you are using an enzyme called DNA polymerase, which is called as TAC. Actually, TAC means it is isolated from a bacteria called Thermus aquaticus, which lives in hot springs. So they live in a temperature around 95 degrees. So a biologist has studied what is there. So basic science is always important. If that biologist is not studied what is there in that hot spring, he won't know that this identifying that bacteria would create even today we are doing all COVID diagnosis being only by PCR. No other technique is, even there are some ELISA antibody based technique, nothing is reliable. Only PCR is even today is only standard gold mark method for identifying coronavirus. So the microbes play a key role on different places, even whatever insulin we are getting or being produced in the microbes for diabetes patients. So if we want to speak about what the microbes are really doing, it will go on for days and weeks and years. So I am going to concentrate on just two issues which would be really interesting, which are the hot topics in the world today. So the first one is I want to talk about locust swarms. So I have to say, this is not a new one. Even in history, there are reports that swarms have came and destroyed the agriculture. So they are now today as evolved as one of the major threat of global food security. So they lead to poverty. So it's not a simple thing. There will be swarms, and generally they are desert based. They originate in desert environments, these locusts, like grasshopper like insects. So they take this path from Kenya, they travel to Middle East, Saudi Arabia, Iran, Pakistan, and now they reached almost North India. Now they are in Rajasthan, Madhya Pradesh, and UP they have reached, and we are not sure whether they will reach Tamil Nadu or not. Even maybe, but we are not sure. So people largely see it as a problem caused by an insect, right? And even there are reports that what we have did is replicating on us. We have killed a lot of birds which feeds on the insects. Insects, since we use for pesticides, the birds are the first target. So that also could be a reason. The global climate change, everything has been known to be a causative factor for this global swarming. So even global warming is a causative factor for this global swarming. So this is how the mechanism, the biology happens. So generally these insects live in solidarity. When they live in solidarity, their body size is relatively large, their wings are small, and when there is a drought or when there is a desert condition, some metabolites, hormones, pheromones has been secreted, which converts them into, which is called gregarious phase behavior. That is, they form along communities and they reproduce very easier, and they, upon reproduction, the infants were with a small body and bigger wings, which make them easier to travel hundreds of kilometers within a day. And this was made by the hormones which are secreted, and this is known in before itself. But later studies have revealed that these hormones or the pheromones, which are some signaling molecules, are produced by the bacteria which are living inside the gut. So the bacteria produces these hormones, and some bacterial species have been isolated from the intestine, the fecal material from these insects, and they have shown the bacteria which are living in this solidarity behavior insects are not producing these hormones. But those which are living, so this bacteria, that conditions due to drought or the conditions are inducing that bacteria to produce that hormones which cause this swarming. So in one way, the bacteria are responsible for this global poverty which would be induced by this food security threat. On the other way, the microbiota themselves provides a solution. There is a fungal parasite. So parasite is a term which is generally refers to the microbes which cause diseases. Generally, any beneficial microbe is not called as a parasite. There is a fungal parasite which also lives in the microbiota, in the gut of the locust. And when this infection happens, it belongs to Microsporidian group of fungus. And when this infection happens, this the hormone production by the bacteria is not produced. So it's interesting. So it's a natural parasite. You can see they have taken a control locus, uninfected and infected locus. After 16 days of infection, so there are three groups of insects. So the blue one is a control insect, and the green one is the insect which are infected. So just see the first and the last bars. So you can see in the control insects, after 16 days, they are uninfected, so they produce these hormones. So hormone serotonin level is measured. They produce these hormones. As a result, they form the grouping behavior. But when it was infected with that fungal parasite, they are not able to. And this red bar indicates they are not really infected. They are just exposed to the metabolites produced by the infected locust. They just collected some fecal materials, and it was done in a closed environment, closed setup. So they just put the fecal materials from infected insect into a box where the uninfected insects are living. And you can see just the exposure to that metabolites itself lead to reduction in the serotonin. So you can see which is shown in blue color is the process done by the microbe. But without this, it cannot produce this metabolite, dopamine and metabolite. So when this fungal infection is there, they kills the bacteria. So there will be a competition. When it is any environment, there will be always competition. When a new person comes, there will be always the survival of the fittest. So when this fungus comes in, this bacteria are not, this specific bacteria which produces this metabolite are not able to survive. So this, if that bacteria is killed, it is not able to convert tyrosine into this dopamine molecule, which is being converted into other substances. So they plays a role in this locust swarming. So this is just one example. They plays a role in everything. So now I come to today's hot topic all over the world, COVID-19. So as I said, they are the drivers of the human life. Yes, they have driven us, and they locked us inside, which we not even thought of even in the last 100 years, even human history, this would be the largest lockdown, and this would be probably due to the development. Even before in the human history also there have been these pandemics have been, but it may be contained to certain regions. But now it's spread all over the world due to development of transport systems and many things. It got easily spread. But when it was during 100 or 1,000 years before, if there is a pandemic, it may be contained only in China, or it may be contained only in India. So in the history, this would be the major event. Even in the abstract I have mentioned, this would be the largest biological disaster ever happened in the human history since their origin of this earth. So it's not just a biological disaster. As you read, as you understand, as we experience day to day, they are making economic disaster. They are doing even cultural disaster. Even many political disasters is also happening, which I don't want to comment here. So several things are happening. So it is caused by so this disease is termed as COVID-19 as the first report. It's not in 2020. Earlier in December 2019 itself, it was identified. So it is called as COVID-19, and it's a type of virus. It has RNA as so the DNA is the genetic material generally in plants and animals, and some viruses has RNA as which is one of the RNA virus, and it was has a lipid layer, and these are the proteins which are called as spike proteins. Since it looks like a crown, it is called coronavirus. Actually, corona means crown. It's like looks like a crown, and not all the coronavirus are serious. There are several types of coronavirus day to day which are responsible for our common cold we are experiencing, and these are a new coronavirus which are evolved. So whatever we are doing, we are getting back. So these, as I said, they are living everywhere. Sometimes the destruction and what we made make them to enter into humans, because they are called as zoonotic parasites. So these diseases are called as zoonotic. They are transferred from different organisms. It could be bats or anything, but bats are not the villains here. They are not. We can't blame them. We destructed their environment. We caused. We disturbed them. As a result, the microbe is entered into the environment, and they very fastly undergo evolution. That's they change their shape. They change their genetic material. Even for a common flu, we don't have a vaccine still date, a reliable vaccine. Even for influenza is one of the common flu which causes disease. We don't have vaccine. Even if you say the vaccine schedule for a diseases which is caused by a bacteria, a vaccine, a one-time vaccine will work for your whole lifetime. For viruses, you need to have booster doses every 3 years once or 5 years once. So it is hugely tough. It is not. Even I am not sure in how many days we will end up with a vaccine or a cure for a coronavirus. But now I want to discuss what's the role of microbes here. Yes, virus is a microbe. Obviously, the microbes causes this disease. Even as I said, virus is microbe or a non-living material is still a debate. We can't. It is in the intermediate phase between the evolution of non-living components and the living component. But still other microbes also playing a role. So I want to discuss two aspects. So what is the role of microbiota? So the disease is called as COVID-19 and the virus is called as SARS-CoV-2. Because already a few years back there had been another kind of coronavirus which is called as SARS, severe acute respiratory syndrome. So they target the respiratory system. So this being a similar virus but with some changes, so they called that virus as SARS-CoV-2. So I want to discuss two aspects of involvement of microbes on this. The first one is what is the role of microbiota residing within us on infection to this virus, and the next one, which was most worrying to me, what would be the effect of disinfectants on microbiota. So first we did some research work which is not published yet, which is just a preliminary data. So as I said, these microbes are infecting the respiratory system and they are causing some breath failure, respiratory arrest, which lead to the death in the COVID-19 infected patients. So microbes entering the lung is not a new phenomenon. Already millions of bacteria, even viruses are living in all throughout the passage from the nose to the lungs. So they are living everywhere, as I said, and they plays a key role. And it is known that there are some microbes living in the intestine, send some signals to the lung, which regulates the pulmonary health and which responds to the disease. And so far, there has been strong data how COVID has been did. As I said, we call them as common cells. So some bacteria are living inside. They don't do harm or they don't do good thing for us. So they are called common cells. But in large, we consider many of them are common cells, but really they are symbionts. So symbiotic bacteria, microorganisms are those which gets benefits from us and also gives benefits for us. So there are a lot of microbes living inside us. And as I said, one of the major benefit is we get immunity. Okay. So even I say you should allow your kids to play with, they should get exposed to everything. They should play with soil. But in the modern society, when we were kids, we were allowed to play with in the soil. But now parents are in the knowledge of something called infection. They say everything is infection, and they prevent the kids from getting exposed to things. So as I said, even common flu was also caused by some viruses which are similar to viruses which like corona. So if a kid was already exposed to that, so immunity is vaccination or immunity is a kind of defense rehearsal. So you, for example, for a bacteria, for a tuberculosis, when you give vaccination, you are giving you kill the bacteria, or a bacterial protein is injected into your body. So you give a training to your. It's a defense system we have, just like any defense training. People who undergo training. Vaccination is a training process. So you showcase to the system that there will be somebody like this will come to you. So you see them, this is how they will look like, but actually they won't cause disease in vaccination, but you show them how they looks like and what they triggers and all those things you show to them, and you made the system to respond to them. Similarly, nature also do a lot of, it may not be call it as exactly our vaccination, but in terms of as they resides, as different kinds of bacteria resides within us, they gives us a lot of immunity to us. And even I was worried with the advertisements when I see even in your paints in the walls will not allow microbes to grow. I was worried. It's not necessarily, it is not all the microbes are harmful. Now I see a lot of advertisements or products which promotes 100% germ-free. 100% germ-free is not good for your health. So if you are not exposed to them, you have no, even there are studies which show that people who take a lot of antibiotics are more susceptible to viral infections. There is no data with this COVID-19, but there are studies in with some previous diseases where they have showed. And also influenza virus vaccination has caused changes in the bacteria which are residing. And even these bacteria produces some metabolites that prevents the pathogens. So as parasites or pathogens are disease causing agents. So good bacteria, Corynebacterium, produces an antibacterial peptide that stops the streptococcus, a disease causing bacterium, from developing a disease. Even there is a study, not only everywhere it is there, even there had been an epidemic on a viral disease in frogs in forest, in Amazon forest. And they have seen that forest in some community or in some regions are getting killed with this virus, and some regions they are not getting infected. When they see what is the difference, they see that the microbes which are living on their skin are different. So the microbes, the frogs which are not killed have some microbes which prevents the viral infection. So they plays a major role. So I was interested whether we are seeing a huge variability with this COVID-19 disease. We see patients, not everybody is getting infected. Even a husband was infected. The wife who stayed with him for some days was not infected. We are seeing somebody is recovering and not others are not recovering. And there is somebody is getting to death phase and while others are not. So there is a huge variation. So we thought whether microbiota plays a role, but working with these viruses is a huge risk task, and even it was even a surprising factor all over India, we have some 3 to 4 facilities only, which is, we call it as BSL 4 or biosafety level 3 or 4 facility, which is present even in the medical colleges, in the government medical colleges. I was surprised to know nobody has that facility to culture and grow a virus. So I was left with, I don't have facilities, I don't have permission to work on the virus. So, and also I want to say, during influenza infection, there has been a lot of studies which showed not really the virus is killing. When the influenza infects us, that leads to proliferation of pathogenic bacteria like Staphylococcus and Streptococcus that remains dormant when they are healthy. But when this viral infection comes, as I said, there is a competition. This virus kills some good bacteria. As a result, these bad bacteria, I just call it as good or bad, which are not scientifically correct, beneficial or disease-causing bacteria to get more proliferated, and they cause the disease. So it is well known in case of influenza, 50% of mortality are due to bacterial pneumonia or bacterial infection. But in case of corona, still we don't have a clear data. As per data I have gathered, it was just 10% maybe they are saying could be due to secondary bacterial infections. But still it is not very clear whether bacterial infection plays a role or not. So at this junction, I was largely suspecting, as I believe microbes are the key drivers of life, I was suspecting microbes play a key role here. But I don't have facility, I don't have permission to work directly on the virus. So as I said, I thought they may be playing a role on the morbidity and mortality, and the viral infection may change. And even there are some, because since we don't have exact drugs known for this COVID-19, different antiviral, even some antibiotics like azithromycin or showing some positive effects in some patients. So could be this bacteria may be playing a role, I was taught. So at this junction, a group from a scientist called Tenover, his lab is called Tenover Lab from US, has published a paper where they used this animal model called ferrets. They are closely related to cats. Actually, generally we use rat or mice as the animal model for diseases, but mice and rats are not easily infected with these coronaviruses. So these are called ferrets. They look like cats, and they have did some experiment in this ferret model with the new viruses. They have isolated the virus from the patient who was just infected 10 days before in US, and they have taken the virus from that patient and they infected in these animals, and they also maintained some animals infected with the other group of influenza virus, and they studied only what changes happens in the small portion, as I said, just 1% of the genes, right? The human genes, in this case, in this animal gene, what changes happens in this 1% of that animal genes. But they did a next generation sequencing, which will give you everything. So it will have all the microbial and all the genes, but they not studied those. They just focused on this genes, and they showed what kind of immune response is happening on comparison to SARS and influenza. But the good thing is they have deposited data in this public database. Now you have some public database, and it's a mandatory. When you get funding from government bodies, you should deposit the data. It's also kind of so that everybody will get help from that. So in that way, they have deposited their sequence from the animals which are infected with these viruses. And I have downloaded this data, and I have looked for, it is just like they have discarded the data as waste. They're not interested in that. I was just mined into the waste, and I looked for, is there any change in the microbes? So this is how they have done. Just so they infected with the thing, and then they collected the nasal wash. They just add some PBS into the nose, and then they collect the wash. In reality, we are doing some oral and nasal swabs. Just you have cotton at the end of a stick, and you take swabs, and then from that only we are now diagnosing the disease. And in this case, they take the nasal wash. And in case of control and this SARS-CoV infected, they did at different time points, and they did sequencing, and they submitted in the database. Actually, these are all not even in the human genes, or in the ferret gene also, they have said it was entirely different between influenza and SARS coronavirus infection. And we are seeing a similar thing there is no big change when coronavirus was infecting, but you can see when IAV is the influenza virus, there is a proliferation of this gamma proteobacteria, which has a lot of infectious bacteria, and there is a reduction in these bacteria, bacteriocytes, chlamydia, and actinobacteria during SARS-CoV infection. They include some beneficial groups also. So in case of, and for this comparative study, we have only day 7 they have did for influenza. So we can compare what I have presented is on day 7. So we can see that it is different. It is not the same how influenza virus caused the disease, change in the nasal microbiota. Coronavirus is doing a different thing, and it is not also that much large, but there are some significant differences where we see some bacteriocytes. You can see this purple color is getting reduced as in case of influenza infection, and this chlamydia is also getting reduced. So and also they have studied at different time points. So these are the abundance of different bacterial classes they have presented in the phyla level. These are the order level. So in the taxonomy we have different orders, and you can see there is an inverse proportional. The bacteria which was, for example, this Burgdorferi is getting reduced when influenza is infecting, but it is getting increased when corona SARS-CoV is infecting. Similarly, in case of this chromatiales, it is increased in case of influenza, but there is no much change in case of corona. Similarly, in case of enterobacteriales. And we see an inverse correlation. Even if you see in case of Burgdorferi, what decreased is increased here. In case of pseudomonadales, is increased during influenza infection. There is no big change, or there is a little bit increase which is statistically significant during coronavirus infection. So we see a huge variation in the microbiota when it is between influenza and coronavirus infection. And these are other bacterial orders, and everywhere we see an inverse correlation. That is what we want to highlight here. And we studied how it happens. So in case of coronavirus, they collected nasal washes at different time points from the days of infection. So after 1 day after infection, 3, 7, and 14 days, and we see a gradual decrease. Or these are the bacterial orders which showed a gradual decrease. You can see different bacterial groups which shows a gradual decrease. You can see in case of pseudomonadales, it was not that much decreasing up to day 7. From day 7 to day 14, there is a huge decrease. You can see in case of here, we see only day 7. Here we don't, in case of pseudomonas, we don't see a big change. But at day 14, it comes down. So what is increased during influenza is getting there. So that, as I said, there is an inverse response between these viruses. And it was getting reduced. These are the different microbes. There is a progressional change. It is not happening just like that. Even in case of Pseudomonas, it was a huge fall down from day 7. But right from infection starts, there is a slowly it is getting reduced. Similarly, every microbes, even some microbes are getting immediately reduced, and chlamydials is reduced, and they are recovering. So these are the microbes which are getting increased. So you can see there is just like a decrease, there is a progressive change which I want to highlight. So it not happens all of a sudden. So as I said, these are the summary of the key inferences which I have explained. There is a inverse correlation, and the changes are different between influenza and SARS-CoV infection. And the one which is increased are they getting decreased here. And it is also important to note that these bacterial orders, lactobacillus, chlamydiales, flavobacterials, and they are largely made up of contains beneficial bacteria, and they are getting reduced during SARS-CoV. And this made me to think whether these bacteria is playing a big role, whether the persons having this bacteria are able to easily get recovered. That's a billion-dollar question. So the reduction in this bacteria causes the health, we don't know. Similarly, we have analyzed some human data also, and we have seen a similar kind of thing. And one of the major limitation of this, so you can see healthy controls, this Corynebacterium, as I said, Prevotella, these are all some beneficial bacteria getting absent when there is a viral infection is there. And the bacteria which are getting predominant during influenza and SARS-CoV are different. So one of the major limitation of this study is it's not done. The experiments are not, the real experiments are not done by me. I just collected the sequences data which was submitted, and I just did some computation bioinformatics study. So even I don't know in case of patients what is their clinical details, we don't have. So I am not able to correlate whether the patient, whether the some events there is a decline, what happens to the ferrets or the humans, they are able to recover. So these things we don't know. So that's a major limitation of the study. Irrespective of these limitations, this preliminary, some bioinformatics study have clearly showed that some changes are happening in the upper respiratory tract due to this infection, and especially some beneficial bacteria are getting reduced, and changes happens progressively, not immediately. And is this progressive changes happens is a key cause for determining the mortality or a infection to a person is a question. Now we are waiting for the permission and grant from the government of India to work on the human samples to see how microbes of different, as I said, the same family, one person is infected and the same person has been in contact with the infected person but not developed infection. So is the microbe residing within us or different, or do they have some protective microbes which helps them not getting infected? Similarly, the patients who get infected, they are, in case of Tamil Nadu, it was nearly more than 50% is getting recovered. And while in case of other states or other countries, the recovery rate is very low. Or due to some food habits or the environment, here we have good nasal microbiota, or due to the air pollution in Delhi, they have a very bad nasal microbiota, which leads to more death. We don't know. This is just a hypothesis based on our preliminary data, but if it really plays a role, even if he knows that some good bacteria is getting reduced just by supplementing them, we can save a patient. So it's a long way to go. We are waiting for the consent from ICMR, and this paper is under review in a journal. So this, as I said, this may help us. So if we know some pathogenic bacteria is more abundant in patients who are more undergoing severe conditions, or who are entering the death phase, maybe we can develop, use some antibiotics which kill those bacteria specifically. Because killing everything is not a solution. That's what I want to highlight here. Similarly, if we identify some comments of good bacteria are getting reduced, maybe giving that bacteria can be a medicine. So maybe understanding all these things could be useful, but we need that system, and I need the samples, which is a long way to go. I'm sure it's not going to happen immediately, because any biological system understanding and then getting into first proving in animal system and then getting into clinical trials, it's a huge work. And even I facing even in day-to-day life, even from my friends from other backgrounds, other subjects, they used to ask me, what you guys are still doing? The coronavirus has came 3-4 months back itself. What you are guys still doing? They used to blame me. But in real life, it is not that much simple. It takes a lot of hard work. It involves, not a single lab can do everything. It involves many big people from all around the world. And it not happens overnight. It takes years. Even I showed the data on that the pesticide causes diabetes through gut microbiota. It is nearly 4 years of work. And it's not that much easy. A biological system is too much complex. We can't simply identify one thing and say just giving this will recover you. Every human is different. So we have to understand everything. The ones which work in the animals may not work in the human system. Even the microbes present in the animals will be different because what they exposed, as I said, the people in Delhi may have a different nasal microbiota as they have more air pollution compared to people in South India. So different parameters are there. So it is not easily we can get, maybe a herd immunity could be the solution, but how long it will take and when it is really a question. So we have seen how microbiota, still we not proved that. We are proposing that microbiota may play a role in a person getting infected, a person getting recovered, or a person getting into the death phase. The other important thing which I want to discuss and I want to stress here is as a preventive activity to get rid of infection. Now we are using a lot of disinfectants. Both government agencies, WHO, everybody is promoting use of hand disinfectants, and we are using masks to prevent the infection. But in other way, they are destroying us. Even I was surprised to see chlorine, the bleaching powder and chlorine are using everywhere in all the surfaces, on all the streets. Actually, whether they will kill this coronavirus or not, even I was not sure. It may be, but surely it is going to kill a lot of beneficial bacteria that lives in the soil, and not only in the soil. We use a lot of soaps and everything. It's getting into the water bodies, and obviously then it reaches the rhizosphere, and it reaches the food, and it comes to us. So everywhere now we are making a huge destruction to the environment in the name of disinfection agent COVID-19. What are the disinfections we are using are not specific to coronavirus. They kills everything. As I said, even there is no solid proof they really kills COVID, but there are proofs they are really bactericides, whatever we are using. So I was very much worried. So I was just starting this discussion. Even I was thinking of starting a debate on this, whether making sterile may be clean, but they are not healthy. If you are washing hands continuously for 10 to 15 times with soap and different sterilizer, even these hand sanitizers, if it is just alcohol, it is fine, but they come with the different chemicals which are proven as endocrine disruptors. There are some things which are present in hand sanitizers which were banned in US but still being, and European countries, but still being used in India, which are well known to cause different diseases like diabetes or heart diseases. And now we are using a lot, and we are also releasing into the environment. Even if I don't use, also in some way through the water or through my food, I will get that. So what would be the post-pandemic challenges we have? Not only health, ecological challenges, because we are destroying all the microbes in the ecosystem. We are just simply spraying the chlorine, hydroxychloride everywhere, and what impact it has, we really don't know. Maybe can create some problems severe than this COVID-19 in the future, which really we have to. There should be a large global discussion has to be initiated to understand what changes are happening when you are daily washing the hands. Even I have. There is a survey. You will get some questions at the end of this talk. Please respond to it. I want to know people who are washing hands continuously, whether they are getting some digestive diseases, whether they are getting some due to continuous use of masks, they develop some breathing problems. So these things are entirely not known. But at this situation, I am not advising you people don't wash your hands. We are left with no options. So it is better you wash your hands. But we don't know. As we left with no options, we have to go with this. But in reality, at least we should reduce avoiding using a lot of disinfectants. Just wash your hands with soap and water or alcohol. Try to avoid chemical disinfectants. And I was also worried about spraying of this hydroxychloride, chlorine and bleaching powder into the environment. I am very sure there are very less evidences that these will have an impact on the viruses. But they are going to destroy the whole ecosystem, the microbes living in the streets, in the air, and even I see those something called as disinfection tunnels. They are some kind of I was very much worried about all those things. Even now it is banned, I think, but during the last few months, especially in India, in Tamil Nadu, they have created some tunnels in which when the people enter a market, they have to go through the tunnel, during which hydroxyhypochlorite or something will be sprayed on all of us. So what impact? They are well known. They will create some diseases in humans, and also what impact they have on the environment. Even they will lead to reduced immunity in humans, which may lead to development of new diseases in future. So I was very much worried on this. As it was written in Tamil, even it was applicable to RO waters, what we are using today. Largely we are using the reverse osmosis water, and as an example, I never spend even a single paise for water. I still remember when I was studying my school days or even doing undergraduation, there was no RO water systems present. We just used to drink the tap water. And it was just 2 or 3 years that entirely it was changed. Now when I drink the tap water, everybody look at me like an alien. They just see like an alien. Oh, you drink tap water. I was surprised. You drink water without nothing. Nothing is not healthy. That is what I try to say. Yes, water can be a source of some infection, but it is not necessarily always. Even when I travel to long places like Delhi, when I used to go by train for 3-4 days, in every station I will get down, I will drink the tap water. Still date, I never spent a single price on purchasing water, and I don't have the habit of taking a water bottle with me even I make long travels, whether it's international or any local travels. So again, I want to say I'm not advising you guys to stop washing your hands, but make it limited and try to avoid using chemicals. Try to just using ethanol is relatively very good. So with this, Sendil sir suggested me to discuss about other works which are going on in our laboratory. So I have named my lab as Microbiomics Laboratory. So on the title, it may seems to be narrowed. The core concept is to understand the interaction between chemicals and the microbes and their implications on ecosystem and human health. So we have a representation of all different kind of chemicals here, from plant flavonoids, pesticides, even diabetic drug metformin and different microbes. As I said, this picture shows all kinds of microbes. This is a microalgae, this is a chlamydia, this is amoeba, protozoan, and this is a group of microbes. And this is a virus, a bacteriophage, and this virus, and this is a virus, and this is also a microalgae. So we are studying all how it interacts with the chemicals, especially with the natural chemicals as well as with the different drugs. And as I said, the pesticides or other environmental chemicals, that's the core of our laboratory. But we work with every system. We have plans to, at present in a hospital environment, we are working largely with the human system. But we work with the Bhavani River here in Coimbatore. We have collected and we've seen what the changes happens in the microbiota when it crosses the industrial area, and then when it crosses the agricultural field, and we have studied. So we are also looking to how, now as I said, how these drugs act in different diseases, what's the role of the microbiota, because some drugs works for some people and some may not. And now there are data, even the are present inside the cancer tissues. It's not just present in your intestine, even in the cancer tissues are there. And we have plans to work on this. And also we have even this Indo-French project, we plan to develop a probiotic, which is a bacteria as a drug for diabetes. And we are also working on collaboration with some electronics professors from engineering colleges to develop some sensors which can easily detect this microbiota. Because when you want to take it to, we used to call it as taking, because whatever biological science did, a lot of things has been done, but there is a strong barrier from taking that from lab table to clinical bed. So only when you make easier techniques which can be done easily and which can be accessible in the hospitals, we can take it to the clinical side. So hope I have given some strong information that all microbes are not harmful. So I would like to thank KMCH Research Foundation and President of KMCH Research Foundation and different clinicians and collaborators, my students, and funding agencies from different bodies of Government of India, DSTs, by ICMR, CSIR, and my host institute has been very much supportive on establishing the lab. So I thank everyone for your patient listening to me. I hope I gave some new information to you, and it will start a discussion globally. I was very happy that the organizers have got me participants from all over the world. Especially I want this topic to taken over all over the world, and some studies has to be initiated so that in future we can avoid some development of new diseases through microbes or through any other metabolic diseases through the loss of these bacteria. So thank you all, and I want to stress we are not bad. Thank you. So
Thank you, sir. Participants kindly use the chat box for asking questions. There are few questions that have been posted in the chat box. Let me ask one by one. Why the nuclear structure of the virus is always changing even among the viruses?
because that's
Sir, can I repeat?
hello.
Sir? Sir, am I audible?
Yes,
Can I ask the question, sir? Yeah. Why the nuclear structure of the virus always changing even among the viruses?
So the first thing is the virus don't have a nucleus. Okay, just like even the bacteria have a nucleus, but the virus undergoes. As I said, it is not even a living creature. It's just a constituent of proteins and the nucleic acid resides inside us. And maybe the question may be targeted on nucleic acid. And these are all very short nucleic acids, and they are easily getting exposed to the environment in comparison to other genomes which are nucleic acid present in a bacteria or a human system. In case of bacteria, it's so we have millions of base pair, but in case of viruses, they are very short sequences, and these sequences they are being just in a small particle, they are easily getting exposed to different environmental factors which makes infection changes in even there are reports the same coronavirus which was identified in China is not the same which was identified in Italy. There are some differences in the sequences. So the environmental factor, they get easily exposed, and it's a short genome, so they undergo changes.
Okay, sir. Thank you. The next question. What is your opinion on autoimmune diseases caused by human microbes, especially by gut microbiota?
Even there are a lot of reports that even gut bacteria plays a role in type 1 diabetes. And as I said, they play a major role everywhere. Even there are reports that by introducing, even in case of type 1 diabetes, there is a bacteria in Akkermansia meschenphila. Okay, that bacteria is getting reduced during type 1 diabetes. And when you give this bacteria to the mice, it was able to overcome that autoimmune disorder even. So even there are many autoimmune, I'm not aware of all the things, but in case of type 1, there are a lot of studies has been done and they play a major role.
Another question,
Hello.
sir. Can we transplant
Yes.
microbiome from a healthy individual to a diseased one to recover from ailment?
Yes, there are even in my study we have done with mice models. We have done even for human diseases ongoing. It may be on clinical practice, especially inflammatory bowel disease, and we are trying to implement it for. That's what our research is going on to see whether even for diabetes also it can play a role.
Another question, sir. When exposed to infected ones, CKOL is higher in its 16 days post incubation. Means carriers are most affected ones at the end.
So in that case, it was getting reduced. That serotonin level was getting reduced. So when there is a fungal infection, it prevents that bacteria from producing that serotonin. I don't completely understand that question, but what I have shown is when there is infection is present, the serotonin production got reduced, and there have been 3 conditions has been shown. The middle one is the one which are not infected but exposed to that metabolites produced by the infected bacteria.
So another question: how global warming affecting the locust swarming?
So as I said, these first of all, some changes happen in this because I am not an entomologist to answer it correctly. Just with my knowledge, I am answering that desert condition, that climate change induces the to go into, so they lose their food, their feeds. So
Another question, sir. Can you suggest for the origin of natural drugs against coronavirus from the microbial consortium inherited with our internal system? As you say, diversity of microbes with our internal organs.
Yeah, it's just as I said, it is just a hypothesis at this moment. So it a lot of research. It may or may not end with, because we have what research we have done is we have just showed there is some variation, there is some loss of beneficial bacteria, but really giving back this beneficial bacteria will recover the patient. We have to do a lot of research on that. Then only, because at that moment I can't suggest anything immediately. A lot of things.
Another question, sir. This COVID-19 is pandemic, even though they are like flu viruses. They are predominant and shows no variation relating to the temperature. Why?
Yeah, as I said, they evolve. It is not necessarily when this COVID has initiated, even there was that India due to high temperature, but it is not necessarily, as I said, it is the virus. They don't have one virus. It's what the reality. You can see the bacteria. Virus is the, and they replicate. They do something into the cell. It may be a bacteria or a human or a cell. So I don't think that the climate conditions play a major role in this case.
Another
so that's
question,
why maybe
sir.
we don't see initial stage itself. We see parallelly there is an epidemic in Italy and Iran. Okay, sorry.
Okay, sir. When so many deaths in Chennai, what's your suggestion to the present situation, sir?
Sorry,
I am not. There is so many deaths.
Sorry, I am not in that position to answer this question because I am not core. I don't have that much knowledge to answer. These are just some initial research. I am a basic researcher, so I can't give some guidelines to the government or bodies. There is a lot of big people are there who have to frame.
What's your advice to the students, those who are going to face the 10th examination in Tamil Nadu?
I think all these, I don't think I am the right person to answer this. Even I can say some suggestions, but I don't want to make those things. Even I can say something, but those suggestions should be made by a psychologist, by educational expert, people like that. Even just like tea kadaiல உட்கார்ந்து பேசுற மாதிரி, I can also say something, but it may not applicable to everybody. So I don't want to comment on those things like policy decisions or suggestions to 10 students like that. It may right or may not right. It differs from student to student. But one thing I want to say to students, have always confidence in you. That's it I want to say.
Thank you, sir. Another question. Can you suggest some sustainable reforms for rejuvenating the lost microbiota in the ecosystem?
Maybe we have to get back to nature. We should avoid using these chemical pesticides. We should avoid using lot of plastics, whatever chemicals we are getting new. We call it as xenobiotics, man-made chemicals. So that could be one of the way we can get back, restore the microbiota in the ecosystem.
And a final question, sir. Why the time is taken for preparing vaccines for COVID-19 to accurately kill the virus?
That's what even during the talk I have explained that clearly, because any studying the biological system or developing a vaccine is not an overnight job. It takes years, and if something happens after giving it to the humans, that's also an issue. We have to make sure, as I said, toxicological risk assessment, there are several stages. Maybe after giving the vaccine, after some 3-4 years, it may create some problem in that human. So before developing a drug or a vaccine, it has to go through several phases of quality control, and many things are there. It cannot be done in overnight, and anything done in overnight may be not beneficial. It may create in the long term, it may create many problems. Okay.
So one more question. If a microorganism is identified that can biodegrade any waste, what can be done for biodegradation of that waste on a large scale? What is important for large scale biodegradation?
So as you said, we have to identify the right microbe. And as I said, the biofermenters or those kind of things can, even there are already some studies and some even what the sewage treatment plants we have, some microbial degradation plant only. Where we have mixture of microbes, and if we identify some microbes which specifically digest some specific like some plastic or something, have to get all the plastics in one place, and you have to provide conditions which we call it as fermenters or something like that. And if you provide that conditions, obviously if it's microbes have the enzymes to degrade that, it will do. Okay.
Thank you.
Hello.
Yes, sir.
There is no direct evidence has been shown so far.
Hello.
Yes.
From the audience. So, how microbiotas are used in drug discovery by Anita Rani. How microbes are used in drug discovery.
Very, very less. Microbes are not hard. And they study their beneficial activities, but comparing to chemical drugs and plant-based drugs, it was very few studies where microbes or direct microbial metabolites are being studied. Even we are not aware of lot of, it's a huge rich resource of microbes which are residing. Very, very few, I would say.
So the next question is, what is your opinion on autoimmune diseases caused by human microbes, especially by gut bacteria? By Kanickai Raj.
I already answered the question, sir. Any more questions from YouTube chat box or somewhere?
Yeah, sir. One more question. Viral strains of corona in US and India have shown variation in their virulence. Medication is remaining the same. How are they provided? Is it safe to use the same for all strains of SARS-CoV-2?
That's what there is variation, but still there are SARS-CoV. There are still some variations are there, but still they are SARS-CoV-2, and there are very few percentage of changes are there. So maybe any vaccines or any medications developed should work for all the different kinds of viruses, of coronaviruses.
So one more question. How to control the locust and their effect on environment?
That's also complex ecological process. Maybe not a single effort will prevent that. It may be it starts from global climate change and maintaining the ecosystem with the bird community and also this microbiota and this parasite infection. So it may not be we can't answer with by a single shot. Maybe we cannot solve this solution. It involves multiple factors from all the people from all different sectors of the society.
Okay, sir. Another question. How the COVID infected people cured? Every day the government report says that same numbers of people cured. How? is by self-defense or by their Medicare?
Even I'm not sure whether that data is not clear. Whether the people, even as I said, in case of Tamil Nadu, by yesterday data, it was around 56% was the recovery rate. But how many of them are treated and how many of them are just been in isolation? There is no clear data on that. I hope majority would be the record based on their some immune response created from their body. Maybe the medicines may played a little or this is just my thought. I am not sure. There is no clear data on this case.
Okay, sir. One more question. Is there any possible condition we can use microbes against locust?
Yeah, could be this. As I said, this if we have, I don't know whether it is possible, but if you be able to infect the locust with that fungal strain, maybe that could prevent. But it was not tried. A lot of insecticides are again being used to kill the locust, but they are not responding to that insecticides. Maybe this fungal parasite may work because what I have presented is the experiment which is done in the laboratory condition. So it has to be taken to the field level, and we are introducing a new parasite into the environment. So we have to make sure that that parasite doesn't cause us any other new problems. So everything has to be controlled. But it's quite possible that, in my view, microbiota can provide a solution for everything because they are everywhere. That's why I'm strongly saying they can provide a solution for everything.
So final question. Why don't we study about evolution from these viruses, from where we can get the clear information of evolution and modifications?
Yeah, there are a lot of studies has been done. If you go to literature, when if you just type in Google, there is a lot of literature is there. Already there have been a lot of reports how these viruses are getting evolved and why new viruses are, because in case of these SARS are not known for before a decade. And why, how these new viruses, as I said, zoonotic viruses are getting into the human system. So there has been a lot of studies already it is been there. So if you just go to Google or any literature, you will get all those details.
Thank you, sir.
Okay. There is another question. Can you hear me?
Yes, sir. Yes, sir. I'm able.
How the pathogenic bacteria are associated with the coronavirus? And then second question is, can bleaching powder kill the coronaviruses?
So the first is, as there is no data in case of coronavirus, how many percentage death is happening due to bacterial infections. Maybe the data may be available, but it is not published or it is not made publicly available. But in case of influenza, nearly 50% of deaths are due to bacterial infection. But I was told by some scientists from Indian Institute of Science, in case of coronavirus, it could be just only 10% maybe due to bacterial infections. And bleaching powder, I have explained there is no strong evidence, direct evidence, I can say, to show that it has an impact on, it may produce some cations which can disrupt the lipid layer. There is no study, as per my knowledge, which showed that this hydroxyhypochloride is able to destroy, and it cannot be used in the living systems because they themselves will cause a lot of diseases in humans.
Okay, another question. Is the pathogenic bacteria reproduction inside the coronavirus bacteriophage and cause secondary infections by one doctor?
So I don't clearly understand the question. So they are speaking about bacteriophage. So bacteriophages are the viruses which kills the bacteria. Even there are some therapeutic approaches are being developed in medical antibiotics. You can use, if you find some bacteria is causing infection during COVID-19, you can give the bacteriophage which can kill the bacteria. So bacteriophages are the viruses which kill the bacteria. So it can be also developed as a new therapeutic approach.
So what is the role of bifidobacteria in human system and related to the COVID?
Bifidobacterium plays a major role. They produce some metabolites and they help a lot in COVID. something is increasing. Still, studies are needed to know what they are really doing. So we are not sure with the COVID what the really. We have did some preliminary analysis, and we have shown. That's why I mentioned there are some limitations of this study. We have just shown there is some changes are happening. Whether these changes influence the virus and the mortality is we have to investigate.
Okay, the next thing is how many strains up to now identified as in the coronavirus? How many strains? Because in the dailies they will be mentioning different type of strains. Based on that, the symptoms are varies like that. Do you know about anything?
I don't know the exact number, but even with have been some 15 different strains have been reported by NIV Pune, National Institute of Virology. So all over the world, I don't know the exact number. It may be more than hundreds. I don't know exactly the exact number.
Okay, then how far the PCR will be identified for 15 strains? How far the PCR will be?
Agents, which is uniformly present in all these type of strains. So the PCR the variation happens in some regions, but the PCR, the domain for which the primers are designed for conserved region of the SARS-CoV, which is uniformly present in all the different strains of SARS-CoV. So PCR possibly can.
Okay, so what about the non-symptomatic corona infections?
Can you repeat the question, sir?
What about the non-symptomatic corona infections? Non-symptomatic, because in Chennai, most of the people are not showing any symptoms, but they are positive. Okay, so how far these PCR will be analyzing?
So that's what, in case of asymptomatic patient, if sample get analyzed, PCR, whether the virus is not, if virus is present, PCR will give you the result. Only if it is present in very low quality, you may end up with not having a positive result. If it is very present in very low limited levels, it may be not. Okay.
Then the next thing is, if the viral is infected, cellular immunity is going on in our immune system. Am I right?
Yes. Yes, yes.
Okay. If the virus is infected, cellular immunity will be goes on. So the major thing is T cell mediated immune response will be takeovers.
Yes.
Am I right? Okay.
Yes, sir.
So at that time, you may know that the memory cell as well as it will be killing the particular viral infected cell. How far it is feasible in antibody detections and antibody therapy?
So antibody therapy, before going into that, so as you said, the immune cells, so even the death is caused by the proficient response of the immune cells killing lot of lung cells in the infected lung cells. A major proportion of the lung cell is got infected, and our immune system kills all the lung cells, which lead to breathing arrest and causes death. And in case of antibodies, only the persons who have responded to the infection will have antibody. That's why this the so-called in the news rapid screening kit is not useful, because rapid screening detects only the antibody. So only when a person produces enough amount of antibody, but as you said, the antibody produced from a record person, as we clinical trials have been happening in terms of plasma therapy, can be beneficial. Because
So
already
I
it is
think,
known that person was.
So I think you are saying as cellular immunity as well as humoral immunity also, both are takes place. If the humoral immunity takes place, then only most of the antibodies are produced, not from the cellular immunity, no? Have you got the point?
Hello? Yes, sir.
Then both the immune systems are working. Am I right? humoral as well as cell-mediated immune response. Okay.
Okay, sir.
Sorry, any other role in interferons as well as interleukins, which are the boosters of the antiviral substance, no? Is there any other role in this controlling of COVID?
Not largely promising, it seems. May have some patients it was working, interleukins are being used, but it is not largely promising. Even so far, no drug has been given a promising response for this COVID-19.
Okay, thank you.
Thank you, sir.
Sir, there is a question in the chat box.
Okay, please.
What are the psychological effects of COVID-19 in children?
As I said, these are all out of my box. I am not the right person to answer all this. I can also say something, so I don't want to in my expertise. Sorry. Maybe you guys can arrange for a debate which involves psychology and different people from different subjects, then these things can be discussed. I am not the right person to answer this.
Sir, Dr. Sagaya Satish from St. Joseph's College, he's on. Sir, you can ask the question, sir. Sir, your mic is not. Yeah, you're not audible, sir. Sir, you are not audible. You are not audible, sir.
Our base okay. Okay. Maybe you call to Sadi sir and tell.
Still, it is not audible, sir.
இல்ல நீங்க போன் பண்ணி சொல்லுங்க மேடம் இல்லைன்னா அவர்கிட்ட.
Okay, sir. Okay.
அவருக்கு நம்ம கேட்கலங்கிறதே அவருக்கு தெரியல. He was speaking going.
Okay. Kanickai Raj,
Okay, let it be. No issue. Okay.
No, it is audible, sir. Please speak.
Hello.
If it is not,
Dr. Anisha, can you hear me?
Sir, yeah. Yeah, I can hear you, sir. You are audible.
Oh, no. Okay. I just want to wish him and you all, because for the past 90 days I have watching many webinars. It's the only webinar with a lot of questions. First of all, I congratulate Dr. Velmurugan. His talk was inclusive and covered most of the aspects and spent a lot of time and answered all the questions. And proud of you be the alumnus of our department. And I also thank you, Velmurugan. Keep in touch with us. And hereby thank our head of the department for his support. And my special congratulations to the organizers, Dr. Rajan and Dr. Anusia. Congratulations, madam.
Thank you, sir. Thank you, sir. Thank you.
Thank you, Velmurugan. Thank you.
Thank you, sir.
Thank you. Any more questions are there?
Sir, actually, there are so many questions which have been posted in YouTube live stream. I have asked only few questions, sir, since some of the questions are repeated. So I asked only few questions.
Video on.
Sir, shall we wind up, sir? Okay, sir. Okay. Coming together is a beginning. Keeping together is progress. Working together is success. It was indeed this mantra of working together that has brought us where we are today. It's my privilege to propose the vote of thanks and acknowledge the contribution of those who really worked hard to make this webinar happen. I, on behalf of Botany Department and the entire fraternity of the institution, first of all, I extend my most sincere thanks to the Almighty God for making today's event a resounding success. With His blessings and grace, we are able to make this event what it was. On behalf of Botany Department, I extend a heartfelt thanks to our chief guest, Dr. Velmurugan, who spared time to grace the occasion. Your talk is informative and have enlightened our minds, sir. Thank you very much, sir. Thank you for providing a wonderful venue here. Thank you. A special mention to our respected father, Leonard Fernando S.J. Rector, Secretary, Principal, and Vice Principal, Reverend Father Santiago, for their encouragement and constant support in all ways. I'm immensely thankful to the head of the department, Dean School of Biological Sciences, for his efforts towards the webinar. I need to mention my deepest sense of appreciation to Dr. K. Rajan, Organizing Secretary, for his effort to conduct this webinar. I also extend thanks to all the faculty of Botany Department and the non-teaching staff for the enormous cooperation. We have been fortunate enough to be backed by a very proactive faculty of our institution, Dr. Kanickai Raj, who is well-versed in hosting the webinar. Thank you, sir. With a deep sense of appreciation and gratitude, we thank our students for their untiring efforts. Finally, I'd like to thank the participants for their patience and enthusiasm shown to listen to this webinar. Thank you. Thank you, one and all.
Thank you.
Thank you, Velmurugan, for
Thank you, sir.
thank you so much for your nice presentations and spare your time with us.
Thank you, sir.
As well as Kanickai Raj, sir, I also express my heartfelt thanks to this. I also express my heartfelt thanks. Okay, sir. Okay. Ma'am, I also thank Anisha ma'am for supporting, as well as one of the organizing secretary, as well as the students. There is a party, economy, who is supporting us. So thank you, Ananda. Okay, thank you.
Thank you, sir. Thank you.